Yeah, hello everyone. I'm very excited to moderate this interesting seminar today on synthetic copolymer nanodiscs enabling near-native membrane protein assays. First, I'll introduce myself really quick. My name is Mario Leutert. As you heard, I'm a postdoc at ETH Zurich at the Institute of Molecular Systems Biology, and I've previously completed a postdoc at the University of Washington, Seattle. So my own research centers on the development of mass spectrometry-based proteomic technologies that permit proteome-wide characterization of cellular signaling and protein regulatory networks. Using these strategies, I investigate the wiring diagram of the cell and try to identify and reverse changes that occur during aging and disease. And so myself, I came into contact with Cube Biotech because I've been using some of their excellent products for phosphopeptide enrichments, also for protein biochemistry and proteomics in general. But the people at Cube Biotech do much more than that. They're also experts for membrane protein research, and they provide services and produce consumable in this area. Specifically, I know that they assist in protein preparation, biophysical target characterization, and also structural determination by electron cryon microscopy. Along this line, apparently they offer the largest portfolio of synthetic polymers that host cell lipids, resulting in higher protein stability and activity. Yeah, now if this all sounds interesting to you, you're at the right place because we have Dr. Felipe Merino here. Felipe is the head of Cube Biotech's cryo-EM division, where he oversees several of their structural biology services, and he's also a key part of Cube's R&D team. Felipe became an expert in cryo-EM through his postdoctoral training at the Max Planck Institute for Molecular Physiology in Dortmund, and then a project leader at the Max Planck Institute for Biology in Tübingen. And his combined expertise in atomic modeling and cryo-EM has provided him with a unique skill set, allowing him to tackle challenging structural biology problems, such as those typically presented by membrane proteins. And with this, I'm very excited to hand the mic over to Felipe. Please take it away.
Yeah, thank you very much, Mario, for your kind introduction. So what I want to talk to you today about is basically one of our most beloved technology platform, which is this copolymers to basically study membrane proteins in as near native environment as possible. So as you just heard, we're a German company. We are situated in Monheim. This is in between Cologne and Düsseldorf. We lived here just right next to the Rhine River, right in the creative campus in the city of Monheim. We are interested in protein purification, and our core interest basically is membrane proteins. So we are a CRO, so that means that we are providing services of membrane protein purification and characterization for other companies or academic labs. And during our services, we're usually generating new tools and new strategies for purifying and studying membrane proteins. So whenever we see that something is working really well, right, or we are designing something that is very important for protein purification, for example, we're going to put it in our shop also as a product, right, which is something that you can also go and do in Cube Biotech's webpage, right? You can directly purchase the products that we're using every day to do our own internal research. So basically, our services are centered in, as I told you, in membrane protein expression, purification, and characterization, right? So, and we at the moment are doing everything from the construct design up to basically structure determination or preparation for any downstream applications. We also can do some other assays, like for example, affinity determination to small molecules using thermophoresis. We can do SPR measurements and things like that. So the core of our R&D part is centered basically on, let me show the pointer here. So on strategies to solubilize the best way possible the membrane proteins, then how to purify them, and then to how to make them ready for any downstream application that you might have, not only structure determination. This is of course the part that I'm most interested in because it is the part that I do most, right? But of course we're doing any other sorts of biochemical characterizations of these proteins. Right, so the question is, of course, why membrane proteins? So they are about between 25 and 30% of the human genome are membrane proteins, right? So most of the proteins are going to be anyway soluble proteins, so why concentrating on this very particular set of proteins? And although they are not so many in number, right, they are very important for cellular life. You can see here, for example, in this cartoon, right, they are sitting at the edge of the cell. And this is basically the first barrier that the cell puts with the environment, right? This is how the cells understand what's around them, get signaling from other cells, interact with their surroundings, they anchor to their substrates, for example. So they are very important ways in which cells can know where they are and what to do. And of course, in eukaryotic cells, they are not only at the plasma membrane, but they are also inside in any membranous kind of compartment, including the mitochondria, for example, and things like that. So they control what goes in and out of organelles and the cell itself. So they are quite important. And you might not be surprised that although they are only about 30% of the proteins that a genome has, they are more than half of the targets of the currently approved pharmaceuticals. So for example, you have that 33% of the current drugs, this was in 2017, right? So not so long ago, were targeting GPCRs. Another almost 20% were targeting ion channels. And this is because they are really, really important. But the problem is that they are some of the most challenging kinds of proteins to characterize. So for example, if we start the cartoon here with the cell that I showed you before, so what you would typically do for characterizing these proteins is that you need to isolate them, right, and have them purified. So what you do, basically, you take your cells, you break them most often mechanically with a sonicator, for example, or a French press. Then you get these membrane pellets. So you can get the membrane disrupted, right? And you can collect the membranes, and these membranes are going to have your protein along with all the other membrane proteins that are in the organism. So for purifying them, what you need to do is to take them out of this environment, right, and get them as single units. So otherwise, you're going to purify them. So what we've done for the last, I would say, 40 to 50 years is solubilize them with detergents. So what the detergent is going to do is to completely remove all the lipids that are present there in the membrane and substitute this lipidic environment by a membrane, by a detergent micelle. Once you have that, then you always have to put in your buffers and in all your applications, you always have to have a little bit of detergent present, right? Because otherwise this micelle would fall apart. Then you can purify your protein. And if you think that it's really important to have the lipids around, then you can just take your protein and reconstitute it into something that mimics differently the membrane environment. So for example, you can use MSP-based nanodisc to put lipids back inside, or you can use the old generation amphipoles that is a polymer that would wrap around your protein and substitute basically the detergent, or you can bring them back into a fully lipidic environment and then put them inside micelles. The problem is that even though you bring them back into a lipidic environment, you are always going to have to do this stripping naked process in between, right? So you're going to lose a lot of protein. The detergents are going to interfere with what you're doing later, right? So this works, of course. This keeps being done because this detergent solubilization is very efficient. And some proteins are happy in detergents, right? But many others aren't. You might think, well, maybe we can skip all this process and then go directly to the reconstitution. And this is more or less what I'm going to talk to you about. There are other technologies that can do this in one step, like for example, when you use saposin, but the problem with that is that you still have to put detergent into the sample for the proteins to be scooped out of the protein, of the membrane. So you're always having this detergent around, which we want to avoid, right? So but what I want you to get from this is that at some point your proteins are going to be completely stripped from their native environment, and we usually do not want to do that to proteins. So this is just an example that is also very close to me because this is an example from cryo-EM, right? There's many others similar that you can find in the literature. This was a project that we had together with MPI Dortmund a few years ago. And this was the cryo characterization of an ion channel from Drosophila. This is called SLOW, right? And if you look very carefully at the methods of the paper, right, you're going to find that at some point for the last step during the purification, we had to take the lipids from the insect cells and put them back into the sample in order for the protein to be happy. And if you look in the reconstructions, for example, you see that there is all these magenta kind of densities that were interpreted by Tobias, the person that did the cryo, as lipid densities, right? So actually what happens when you do not take this step and you do not put the lipids back into your preparation, this is kind of the reconstruction that you get. And then you see that in the area where the transmembrane region should be, there is basically nothing ordered. So the protein is really unhappy in just detergent. It needs to have these lipids there. And what we've done like a few years later was to, just as a part of our QC, just checking out that all our cryo pipeline was working correctly, we just took the same sample, purified by a completely different person, reconstructed by a completely different person, and we still see that those lipids are exactly in the same position. So this is a reproducible thing that we see with this protein, right? And these interactions seem to be really important. And of course, you don't have to take our experience only as an example. There is many other papers like the ones I'm showing you here that show you that not only lipids are important for keeping the structure of the protein, they are also involved in maintaining or determining specific functions. Because you're not only going to have specific lipid-protein interactions, there is also kind of a more passive effect of the lipids around your protein that are going to create specific shapes that are going to be kind of tailored to your protein, right? And then you're going to want to have the right lipid composition to promote the correct function in a way. So we have basically this kind of levels of nativeness, if you want, in the different preparations that you can have, right? So the least native, I would say, is detergents. Amphipols maybe are a little bit better, although they would be comparable to detergents. The king of cryo-EM, I would say, is MSP-based nanodiscs, because these give very nice reconstructions, but you still have to go through this process of stripping your protein naked and then putting the lipids back again. What we like better, and this is our core technology here in Cube Biotech, is synthetic copolymers. And in synthetic copolymers, you're going to end up with something that looks very similar to an MSP-based nanodisc, but there's some key differences that I'm going to show you in a second that we think make it a significantly better technology as any of the other available technologies. Right, so they were introduced around 2010, right? As these, they were called MAS. This was the first polymer that was introduced. It's a polymer made by maleic acid here and styrene. And what they discovered in the original paper was that these polymers, so you have repeating units of this to a molecular weight of like 10 kilodaltons or bigger. And then these polymers would go into the membrane and incorporate, and then self-assemble into a nanodisc. And this polymer itself would scoop out basically the protein out of the membrane, but the important part is that they would scoop out the protein from the membrane together with the native lipids of the protein. So this is from a different paper. I think this is from 2014. And then you see that these are basically now thin layer chromatography, not to check the protein, but to check the lipids present in the sample. And then these are different steps in the purification until you get to the purest part, so only your protein, right, in its nanodisc. And then you can see that basically there is different kinds of lipid head groups there. So the lipids are still there, right? The protein is isolated and is relatively happy. So this is exactly what we want. So fast forward to now, and basically these are the four main families of polymers that are available, or the main families of polymers that are available. You have the original SMA, that it's maleic acid with styrene. Around 2014 or 15, the group of Sandro Kella introduced these DIPMA polymers, which is the isobutylene with maleic acid, so you don't have the ring anymore. A few years ago, this new one was introduced, right? That is not maleic acid, but it's acrylic acid. So you have basically less charge in the backbone. And then a few years ago, the group of Manuela Sunens introduced this new generation amphipols that are commercially called ultrasolute. And basically the main difference with the original amphipoles is that these ones have a different side chain here, and they now have the capability of solubilizing proteins just as the other polymers. So these are basically the main polymers that you're going to see in the literature today. So now how would a protein purification would work with these guys? So basically up until this step, for example, is exactly the same. So you collect your membranes, but now you put the polymer inside. And instead of having to solubilize and reconstitute and everything, this is going to directly create native nanodiscs from your protein and all the other proteins that are in the cell, right? And then from this point forward, basically you can think of your protein just as a soluble protein. So you never again have to put polymers inside, detergents, or any additive whatsoever other than your buffers, salts, and the typical things that you would use for membrane soluble protein work, right? And then you can purify your protein, and then you have your protein again in a nanodisc. But the advantage of this nanodisc is that it has the lipids that are preferred by your protein and not the ones that you put back into it. And it was never naked, right? So the idea is that with these polymers, you can preserve the local environment and preserve function and stability of the proteins, which is kind of an issue, especially with membrane protein work. Right? The problem and why these polymers got a bad, I would say, people got the wrong idea perhaps about these polymers is that this is just an example from the literature as well. This is how a typical solubilization try of this is beta-2 adrenergic receptor, for example, but most proteins would look like this. You have solubilization in DDM, and then you have this very fat band, right? You say like, okay, solubilization worked really well. And then you compare it with, in this case, with DIFMA, right? And then you see that the solubilization efficiency is significantly lower than DDM. So many people thought like, okay, these polymers are really not working so nicely. But systematic characterizations of the polymers against detergents are not so common, right? Or they were not done so often, right? So other than individual examples of particular proteins, we don't know really how badly, if at all, these polymers perform compared to, for example, state-of-the-art detergents. So what we did was to say, okay, you know what? Let's do this experiment where we get to this part where we have all the proteins solubilized. So we collect the membranes, we throw the polymers inside. But instead of purifying single proteins, we did this with hex cells, right? So to have an idea of how this is performing on the human proteome, membrane proteome. And then we directly sent it to an LCMS to do mass spectrometry and try to see how many membrane proteins are we really getting out with these polymers. And now we took advantage of the very large amount of polymers that we have now in our shelves, right? So we went to the shelf, we took all the polymers that we had in 2021, and then we tried all of them, and then we compared them against typical detergents, both aggressive and not aggressive, to see really how these things were working out. So, right. So the first thing we saw, like the negative control is just HEPI's buffer is here. Then we have two controls of mild detergents and aggressive detergents, right? So this would be our positive control. And hopefully you would expect that our polymers would end up somewhere here in the middle, right? So we are not counting here right now how much of membrane proteins in mass you're taking out, but rather how many different membrane proteins you can identify in the solubilization itself. And I'm gonna come back to so now this is about the versatility of the polymers, and I'm gonna come back to how efficient they are extracting the membrane proteins in a second. So what was perhaps most surprising, we were expecting these things to land kind of in the middle, right? We knew that they weren't going to be as efficient as detergents necessarily. What happened, so this is the line that is marked here. What happened is that, for example, new generation amphipoles end up basically being as versatile as detergents, both ionic and non-ionic, which was a surprise, right? We have come to accept that these ultra-solid amphipoles are basically the star solubilizers. They are very efficient. But also SMAs are performing really well, and even the ones that have the lowest solubilization capacity, which are DIFMAs, they are still significantly better than the negative control, and then they have a very respectable solubilization capacity. So they turn out to be very versatile. And the advantage here is that if you look at the axis, is that these are thousands of proteins that we're probing at the same time. This is not a single example, but we see that they are able to solubilize thousands of different proteins from a human cell. So what we did then afterwards was we know that they can solubilize several proteins at the same time. So what we did was for each of the polymers, get kind of a solubilization profile. Let's say protein A is solubilized with this efficiency, B with a different efficiency, and so on and so forth for these thousands of proteins, right? And do principal component analysis with that. And we wanted to see if basically the detergents and the polymers were going to end up in different places of the principal component space or not. So in other words, we wanted to know if is there a solubilization fingerprint to these things, or are they all being kind of general solubilizers? And the first thing that was kind of interesting was that when you look at detergents, they all cluster together. Ionic and non-ionic detergents end up more or less in the same space, except for digitonin. And we know this wasn't so surprising because digitonin is a very mild detergent, so it's not necessarily comparable to these other ones. What was interesting was to see the repo buffer here. This is a buffer used for mass spectrometry usually, and this buffer is generally not conducive to native prep. So it's really aggressive. It's going to get everything out right, but at the expense of the structure. When we compared it to how the polymers look like, this was very interesting to see because all the families of polymers basically clustered differently in this space, right? So diff mass end up here, Assays in a different place, and ultrasolute in a different spot as well. So where they end up in space wasn't so interesting to us as to see that the families are kind of clustering together. So it's really that families of polymers have different solubilization fingerprints, basically. And we thought like, okay, maybe this can be due to specific characteristics of the protein. So maybe more hydrophobic proteins are solubilized by a certain polymer better, or more negatively charged are solubilized by a different polymer better. So this is exactly what we checked next. We checked whether these differences were coming from anything that we could calculate directly from the sequence. But as you can see, for example, this is the isoelectric point of the proteins that we are identifying basically in our mass spec runs. And either when we look at the transmembrane regions or the full protein sequences, we do not see any difference in these violin plots for any of the polymers. So I don't think they are almost identical for all intents and purposes. We didn't observe any difference in isoelectric point, nor in the hydrophobicity of the proteins. So the only difference is, of course, between the negative control, which is HEPIs, and the rest of the polymers, but this is kind of expected. Right, so we are not seeing any differences in something that we could kind of trivially calculate from the sequences. So if it doesn't come from these characteristics, then where does it come from? So I think it's still determined by the protein itself. But if it doesn't come from the primary sequence, then it has to come from a tertiary sequence, a tertiary structure, sorry, which means that it comes from the protein structure, right? So what we think is happening is that I borrowed this from a molecular dynamic simulation paper. And what they are showing here is basically that they tried this for different proteins, right? And they had a mixture of lipids that were the mixture of lipids that you would have in a normal human cell. And they saw exactly for different proteins, you have these areas where specific lipids get enriched or depleted around the protein. So this is for fully saturated lipids, this is for polyunsaturated lipids, and this is for cholesterol. And then you see really that in some areas cholesterol is going to bind really tightly to the protein. This is the upper leaflet. In the lower leaflet, you see a different pattern, right? So it's really like the protein is tailoring its own lipidic environment, or there's these specific interactions that are going to fit the shape of the protein, and this is going to end up in a local specific lipidic environment, in a local lipidic fingerprint for each of the proteins. So we think that this is what the polymers are sensing. And unfortunately, this is not something that we could calculate trivially, right, from the proteins themselves. So the take-home message would be really, if you want to find the best polymer for your protein, then you have to screen. But the good news is that you're going to find probably one of the polymers that really solubilizes your protein, and the protein is really happy after solubilization. Right, so up until now, I told you about the versatility of these polymers, right? So how many membrane proteins different membrane proteins you can get out of this of the cell. But of course, we try to calculate also raw efficiency, right? So how much material are you getting out of the cell? And this is basically kind of a measure of the total amount of signal that you're getting from the samples. So you see that, and we did this experiment now, not at saturating concentrations of the polymers, but we really did a little gradient, right, from 0.1% to 2.5% to see exactly which ones were more efficient, right, and how much you're getting out of the cell, and maybe if some polymers really need higher concentrations than others. So you see that for element G and DDM, already 1% is really high, right? And you can see now that with amphipoles, you get a very similar profile, and you see that the scale in all the graphs is exactly the same. So you're getting not only kind of the same saturation, so 1% is enough, but also at 1%, you're getting almost as much as you get in detergents. So it's really not only versatile, but very efficient compounds to solubilize your membrane proteins. We see that SMAs are a little bit less efficient. This also is basically what we knew from before. And some of them really, like this SMA 200, takes a little bit longer to saturate. You need a bit higher concentrations. TIVMAs, I think they kind of saturate at 1% as well, but at the expense that you're not getting as much protein out of it. And ASTIs are also very efficient, particularly the 650, which is very small, so we think it's very aggressive, solubilizing the membrane proteins, and then you get performance similar to here with amphipoles. So this was cool because now we see that they are as versatile as detergents, and some of them are actually as efficient as detergents. So this is how basically the raw data would look like now. So what you have here is a very large amount of data. So basically this is the details of the experiment I just showed you. So we have 5 concentrations for each of the compounds, right? And each row in this heat map is basically one protein. So we have here like 1,800 proteins that could be at least detected in one of the compounds, right? And then we group them also by the number of transmembrane passes that they had. So you can see really that it's not only single pass transmembrane proteins, but you have 2 up until 10. I think the biggest one in this data set is like more than 20. So it's really, they can not only solubilize small membrane proteins, but they can solubilize big membrane proteins. And I think this is one of the advantages, or the main advantages, that you would have over MSP-based nanodiscs. The size of the nanodisc is going to adapt itself. You don't need to use differently sized polymers into it. As you would need different MSPs for your nanodiscs of different proteins, you just mix them with the sample, and then it's going to take out the appropriate nanodisc out of it. So, and what I wanted to highlight here is that you can see that compared to element G or DDM, ultrasolute 17 and 18, and also these ASTI show very similar profiles. So of course, this is a lot of data. If you want to zoom in a little bit, this is how everything looks like, right? So element G and DDM are working very efficiently with most of the samples. There are some proteins that you can even detect in the negative control because maybe they got proteolized during preparation, or maybe they are just loosely attached to the membrane. But you have basically similar to element G and DDM, the 2 ultrasolubles are working really nicely. But you have many proteins here that are not, for example, in the negative control, and then you see them in dimaglycerol, for example, dismaten, some of the SMAs, right? So it's I think we've come to a point in the lab where we think that if we can get a protein expressed in cells, a construct that expresses really well, then we're going to get that protein purified with polymers. So it's the purification of the protein is not the bottleneck anymore, but it's really the production of the protein in the cells. right? So this is a lot of valuable information. So how could you access that? So of course we have the preprint that has all this data, right? But we also have created a solubilization database. So for example, if you're interested in a protein, in particular this one here, for example, you go to our website, to the solubilization database, put that information there, and this is the kind of answer that you're going to get. If we could detect it in our tests, then you're going to get a link to your protein, right, together with the solubilization profile of that particular protein. So you can say, okay, this one I'm going to probably solubilize or try to solubilize with this MALP here, or I'm going to try this dimaglucosamine because I'm interested in that particular backbone or something like that. So the solubilization database, it's available basically for everybody to use. You can just go to the link here at the bottom of the screen, and then you can check if you can see any information on your protein of interest. So right now I just talked to you about raw solubilization, right? So what I haven't touched on is whether or not these proteins are happy, whether or not these proteins are functionally solubilized, which is kind of an important thing, right? So this is basically our workhorse right now. This protein is called P2X4. It's a trimeric ATP receptor. It's related to pain, so it's pharmaceutically very interesting, right? So the protein itself weighs like 120 kilodaltons, and with the nanodisc, it's a little bit more, of course, right? And it's involved in neuropathic pain. So this is the kind of pain that you feel not when somebody hits you, right? But it's the kind of pain that you feel when you have a severed nerve, for example. So it's not so biologically useful anymore, and there's a lot of necessity to drug this. So this is how a solubilization campaign basically would look like with P2X4. So we see that the protein gets very well solubilized with several assays. Ultrasolute 18 works really nicely, and one of the SMAs works really nicely as well. So this is the proteins, and this is the Western blot for the raw attack that we're using always to purify our membrane proteins. And in this case, we decided, okay, let's go on with ultrasolute 18 because it's one of the best polymers that we have. So this is how a purification of P2X4 would look like, right? So you see that this is the control that we have in DDM, CHS. Then you see that the peak is not so homogeneous as you would like. The peak in ultrasolute 18 looks really sharp, really nice. This is what you want to see for a purification of a protein. And then this is some work that we've done together with Edelgries, a company doing ASMS. And this is a silver staining gel, so no, it's not anymore Kumasi. And then you see that in silver staining, you have the P2X4 solubilized in ultrasolute 18. It's really, really pure. So there is nothing else than P2X4, even better than what you get in DDM. So this was very encouraging. So we've tested this in the stability of this of P2X4 in polymers compared to detergents. So this is a temperature ramp that we did in our Panta, right? So this is a nanotemper Panta. So this is a DLS measurement particularly. And then you see that at different temperatures, there is a certain variability in the size distribution of the particle, but it's really not that large difference. When you do the same with the DDM CHS, you see that already at 45 degrees Celsius, the protein really starts to aggregate, and then it's irreversibly aggregated into something that is not useful anymore. And we are very excited about these kind of results because this tells you that if the protein is really stable up to 40 degrees, then maybe you don't need to do your assays at 4 degrees anymore, but you can really do them at 37. And maybe this is something that is really important for you, especially if these things are going to go at some point back into the body, right? And it's important also that we can find this superior stability compared to detergents because some applications are really going to need that. So if you need to raise antibodies, for example, then you want something that is going to survive a little bit longer when you're kind of immunizing an animal, for example. So this is really nice to see. So as I told you, we partner up with this company doing affinity selection mass spectrometry. So there's a few things that you need to do to identify basically compounds for proteins, right? So you are using your protein to pull different compounds through a matrix, right? So this is basically a small size exclusion chromatography if you want, and then you need your protein to go really clean through it. So this is the first thing that they tried, and this was one of the first encouraging results that they got, is that always in all the matrices that they tested, ultrasolute 18 was outperforming detergent. So the detergent seems to interfere very much in the recovery of the protein after passing through the matrix. This was not the case for ultrasolute 18, or to a significantly less degree to ultrasolute to DDM. And when they tried to actually measure the affinities of known compounds, right, then they got, again, these are preliminary results, right, but they got something that was very similar to what is known in the literature for these compounds. This one gets a little bit of unspecific binding there in the end, and this is because there's some issues with the solubility of the compounds themselves, because this negative control should be basically here at the bottom. So it's more of a technical issue than an issue with the protein prep itself necessarily. But this was, of course, very encouraging to see. So we have something that lives longer and it's functional as well, which is good to see. Again, so I'm an electron microscopist, right? So I'm a structural biologist, and then what we try to do also is to find better ways of getting membrane proteins into structures, right? And this is when we found a minor bottleneck. You see on the left, you see the protein in DDM, right? So these are 2D classes of that protein. They all look very nice. When you look on the right, this is the same protein in ultrasolute 18. So two things that you might notice. One of them is that the belt here, so the native nanodisc, is significantly smaller than the belt that you get with DDM. We knew that was going to happen because the particles look smaller in the DLS as well. The problem is that most of the protein comes from the top. So you see these two ones are top views, and then you can hopefully see that the details in the 2D class are very crisp. So the protein seems to be happy, but we're seeing it from a single side, and then you see that in these histograms here. So this is a histogram of how many particles are looking for from a specific direction. So everything is kind of in this direction, where you see in the DDM case, they are kind of around the protein. And this becomes a technical issue for cryo-EM. This is why we could not solve a structure with ultrasolute, but we know that the protein is happy because you see very high resolution details in the classes. So this is how the structure is looking like, right? So the prep is very nice itself. So we have basically a known antagonist here bound to the soluble part, plus several glycosylations around the protein. So this is, of course, work in progress that we're doing right now. But not to get disencouraged by this. So these are all the structures that have been solved by other people, not by us, in the literature with native nanodiscs, right? So you have several cases where you can get even below 3 angstrom resolution. So this is very encouraging. In many of these cases, you can see lipids in the preparation. So this is also very encouraging. And if you can see the dates, this is something, a field that is really emerging right now. So the oldest paper is from 2018. So structures keep coming up every year, right? And then the important thing is that we have proteins also from E. coli, from other bacteria, and from eukaryotic sources. So basically, you can do this with proteins from any source, and then you can, in principle, get high resolution structure for all of them. So, right, the last thing I wanted to touch on before ending the seminar is that maybe some of you thought like, okay, if these polymers really can incorporate into membranes, why do we need to break the cells first and then collect the proteins, right? Can't we just mix the polymers with living cells or with any cell, right? And then the membrane would get solubilized directly. So we ask ourselves the same question, and the answer is yes, you can do that. So basically, this is now with the hex cells, right? So this is ripple buffer, the one that I mentioned before, that usually is used for mass spectrometry. And this is a buffer that we have called gentilized. So it's basically based on our polymers as well. This is 1 minute after mixing with the polymers, and then you see that already. So the experiment was we mix our buffer with the cells, right? Then we pellet everything that is insoluble, and then we run a gel of what's left over, right? So if there is no breakage of cells, then there should be no proteins here, or almost no proteins. But you see that basically there is comparably as many proteins as you have in the riba buffer. And if you incubate this for a bit longer, half an hour, then you get really lots of proteins starting to appear, whereas in the control, right, where there was no compound there, it's almost completely empty. So you can really break cells like that. And as you can see here, so if you count the number of cells that you could see under the microscope, right, after a minute, you already have a significant drop from the control. And after 10 minutes, you have basically lyse more than, I would say, 2/3 of the cells with our gentilized dissolve buffer. But of course, we didn't only make one to break cells, we made another one called gentilized stabilize that breaks the cells and also solubilize your membrane proteins all on the fly, right? So you mix these things with the cell, and then you're gonna get basically your soup of membrane proteins and soluble proteins directly there in a kind of a not native, but not near native kind of environment. And the advantage of this is that you can break your cells, and then you don't have to use this very harsh treatment that you would do, for example, by sonicating them, or putting very strong detergents into it. And the advantage of that would be that if you're after something that is a very labile complex, for example, it would not break apart with gentilize as it would break apart with a detergent-based lysis buffer. And we see this being applied, for example, in the case that you have, let's say you don't want to purify your proteins to do DEL screening, right? But you want to do DEL screening in lysate. So you would take gentilize, mix it with your cells, the cell would open, then you throw your DELs, and then you would purify your protein directly, right? And in a very gentle environment, kind of maximizing your chances of getting a view of the native-ish state of the cell. Right, so we created these two buffers, right? And then you can get them also right now. They are basically the same, except that gentilized dissolve will not extract your membrane protein, so it's only going to be for the soluble part of the cell. But with stabilized, you're also going to get the membrane proteins into native nanodiscs. So with that, I would like to wrap up and tell you that remind you of what I would like you to take home today, right? So I showed you and hopefully convince you that these are very versatile tools to solubilize and stabilize membrane proteins, that they all have their own solubilization fingerprint, right? So it's very important that you screen the polymers and choose the one that best fits your needs. I showed you that they offer improved stability compared to detergents, which is a very welcome characteristic of these polymers. But what's very important here is that more is not always better. So you are not necessarily going to end up choosing the polymer that gives you the best solubilization efficiency, but you have to tailor fit them to the downstream applications depending on what exactly are the needs of the technique that you're going to use afterwards. And these polymers can also be used to lyse eukaryotic cells. So then we were very excited about this new development, and I encourage you, if you have a project that needs this, to try it out. So with that, I would like to thank you for listening to me today. And I would just to acknowledge basically some of our partners, right? So this protein solubilization mass spectrometry work was done in collaboration with CICAD, with Stefan Müller and Jan-Willem Lackmann from CECAD in University of Cologne. And the affinity selection work was done with in collaboration with Eldres. So you can, on the left upper side of the screen is a link to the preprint about the mass spectrometry work. So if you want to have a look, then go there and then please check it out. And with that, I would like to, again, thank you for your attention, and I welcome your questions.
Yeah, that was definitely a very nice, informative seminar. So please put all your questions in the little type box on the right. And also, if we don't have time to answer the question, the Cube Biotech team will later follow up on your questions. Maybe I can start with the first question. So, I mean, you said that you screen like all the different polymers to find the best one for the specific application. Is there also a possibility to just use a mix of all the different polymers, or is that basically?
Yeah, that's a good question. So we've been exploring that. I don't know to which extent you're going to be able to separate them afterwards. So if you have a mixture of different polymers, I don't know if you're going to end up with something that has always a mixture of the polymers or different combinations of native nanodiscs embedded into it. But that's definitely something that we're trying to find out, if whether or not we can mix them to get better solubilization or things like that.
Yep, good. And I was also wondering, so you talked about ASMS. Would it also be possible to use these polymers for co-immunopurification applications, where you basically identify protein ligands of membrane receptors or something like that?
Absolutely, absolutely. So we I think we've done that before. Well, it's definitely possible. It's just, so one of the main problems, I don't think for a co-IP would be an issue. So some tags, affinity tags, don't work so well with polymers. So for example, his tags are not going to work so greatly. But I'm guessing if you're doing co-immunoprecipitation, you're going to be pulling them out with an antibody. And if you do that, then yes, absolutely. And I think it would be significantly better than doing it with detergents.
Okay, yeah, definitely interesting application. Yeah, here we have a question: are there any drawbacks of using copolymers instead of detergents?
Right. So one of the issues with the copolymers is that they have all these carboxylates in the backbone, so they will interact very badly with divalent ions, right? So many of them precipitate after you add, let's say, more than 5 millimolar magnesium, for example. Then you would need to choose. There are several that don't have that problem, right? So you would just need to choose one that that doesn't have that problem. Deep bonds, for example, are very good for that. Any other drawbacks is that, as I told you, so nickel and TA doesn't work really well with them, so you have to choose an appropriate tag. We favor our raw tag because it's an antibody-based purification. It works really nicely. But other than that, I think it's all about tailoring the polymer that you use to the specific needs that you have. So some of them absorb at 280, right? So if you need to measure something at that wavelength in your application, then you should choose a polymer that doesn't do that, right? And things of that sort.
Okay. And then another question would be, there are many membrane proteins that were either missing or very poorly solubilized in the human membrane proteome experiment. Is it possible that copolymers will fail with some of these targets?
Yeah, that's an interesting topic. So yes, we can. So in the database, of course, there's going to be many human proteins missing because either they are not expressed in hex cells, so that's entirely possible. Or what we saw is that when you compare the presence of a certain membrane protein in the mass spectrometry data with the number of transcripts that you have for that particular protein in hex cells, there is a direct correlation. So we saw that the main issue with identification of proteins in our hex cell experiments is that the proteins are just not in high enough copies. But for overexpressed proteins, like you would do for a protein purification, we have never encountered a single example where this is not solubilized by a copolymer.
Okay. Yep, please keep on asking questions, put them in the chat. Here we have another one. What should I consider when choosing the appropriate copolymer for a specific application?
Right, so as I was saying, for example, if you need to do something analytical for calcium binding, for example, then you would take one of the polymers that doesn't bind or doesn't interfere with the valent ions. Or if you need very strongly to measure at the 280 absorbance, then you would take something that doesn't have a styrene, so it doesn't interfere with your measurement. So, but it all depends. Maybe it's always good to have a few, after you find out which one solubilize the best, then to have a few that you try against your technique, and then you say, okay, this is one that's giving me the best result.
Okay. And I was actually wondering, is it possible when you have your proteins in these nanodiscs with your polymers, can you freeze them and for long-term storage and reuse them later?
I guess this is dependent on each protein. So some proteins are going to survive that better than others. We haven't seen a specific case where a freeze-thaw cycle would end up in the protein being kind of a denature or something. But I guess this is something that you definitely need to try per project. I wouldn't do it blindly.
Yeah. And one question here in the chat is, would the gentle lyse buffer work on other kinds of cells?
Right. So no. Well, yes. So it it works in animal cells. It definitely works. So we have seen it working in tiny cells, so insect cells, and on hex cells. These both work equally well. It does not work for bacterial cells.
Mm-hmm.
So if you have a eukaryotic application, then definitely it works. If you have a bacterial application, we're working on finding a solution for that. But for the moment, it does not work for bacterial cells.
Okay. And one more question that I had for your big proteomic screen where you tested the different polymers. Could you see any enrichment for different membrane types? For example, some polymers are better with ER membrane proteins or something like this.
Yeah, that's a good question. This was one of the things that we wanted to explore. So because if there is a specific preference for proteins for the lipidic environment of each protein, then you would expect that maybe some polymers are better for mitochondria, for example. Some are better for Golgi. The problem is that since we were working with total membrane lysates, I don't know to which extent the membrane asymmetries were kept. Because you break the cells and then everything is pulled together, and then everything is solubilized in nanodiscs, and then the nanodiscs themselves can exchange lipids. So I think there should be an effect like that. I don't think in our experimental setup it was possible to see that.
Okay. And maybe one more question. So were you also planning to do any lipidomic experiments that maybe the polymers capture different lipid types?
Yeah, so this is definitely in the pipeline. So there's some other people that have done that. You can basically, for each of the proteins or for each of the particles that you have, you can try to find out exactly which lipids come with it. You can see it in other people's experiments that you get different lipid compositions for different proteins, right? But we haven't done the experiment yet. Definitely one of the things that we want to try, because it's important to see exactly that the proteins come with their lipids, and it's really its native environment.
Okay. And then in general, for these nanodiscs, are they pretty uniform in size, or is there a pretty big spread?
So when you make nanodiscs from just membranes, the size of the nanodisc is more or less uniform, and it depends on the lipid to polymer ratio. So you get different sizes depending on how many lipids per polymer you have. With the proteins themselves, I think to a certain extent the protein dictates the size of the nanodisc that you're getting. I think a few polymers are gonna kind of arrange in a way that it gives the size that the protein needs.
Okay, so the assumption is that usually you capture only one protein within a disc.
Yes. So within one complex, I would say. So something tightly bound, I guess you by chance are gonna end up you could end up with two proteins in the same nanodisc. I think doing purification, you're gonna get rid of these things just because they don't have the right size, and usually there is a sizing step in the purification process. But I've seen that as well in cryo-EM, sometimes in MSP-based nanodiscs. So even though they are supposed to have a fixed size, you sometimes have two proteins inserted in the same nanodisc because you cannot really control that, right? That it happens every now and then.
Okay. And do you think you're done with finding new synthetic polymers for these types of applications, or is there other type of polymers that still could be discovered?
We are always incorporating new polymers into the portfolio. So very recently, we made a collaboration to introduce a new type of SMAs developed by Bert Klumperman, for example. Yeah, so they keep coming, modifications to the backbone or modifications to the side chains and make them more or less hydrophobic, right? Or that they are neutrally charged instead of negatively charged, right? So these ones have proven to be very popular lately. So definitely improvements are to come.
Okay. Yeah, I don't see any more questions for the moment, but yeah, if you still have a question, put it in there and the team will follow up with it. And I think, yeah, if you don't want to say anything anymore, Felipe.
Yeah, so just to thank everybody again for attending. And then if you have any questions, just drop us an email and then we're gonna do the best to answer them.